Hi dear students, now we are going to talk about another type of microbial interaction called the cooperation or the proto-cooperation. A cooperative relationship is also a positive relationship which is happening between two different organisms, but it is not found to be obligatory. That is, it is not a must reaction. And for most of the microbial ecologists, this non obligatory aspect differentiates the cooperation from mutualism. In the case of mutualism, we know that the symbiotic relationship is a essential factor for both of the organisms. But in the case of cooperation, such a must factor or an obligatory factor is not being present when two organisms are being found to be associated. And unfortunately, it is quite difficult to distinguish between obligatory from non-obligatory because which is obligatory in one habitat may not be the same in another. So, if you go to consider in a natural environment and in a laboratory, the demarcation of saying which is obligatory and which is non-obligatory, that will be a bit different. But we can, in spite of all these differences, we can say that the most useful distinction between a cooperation and mutualism is that the observation that the cooperating organisms can be separated from one another and still they can remain viable as well as functional if in the case of cooperation. So in the organisms which are being involving in a proto-cooperation or a cooperative association, you can see that even after they have been separated from one another, they will remain viable. And not necessarily always that they will be functional, but they will remain viable even after they are being separated. But in the case of a mutualistic or a symbiotic relationship, you can see that once they are being separated, they will not be viable as well as they will not be functional also. So that is the case of proto-cooperation or the cooperation. Now, if you go to take this proto-cooperation as well as cooperation, you can see that they usually involve syntropic relationships. What is a syntropic relationship? If any association will permit the growth of one organism or will either by improving it or by promoting its growth by providing growth factors, nutrients, or substrates, which is being by an organism which is growing nearby, then we say that such an association is referred to as a syntropism. That is, when two organisms are being what associated with each other, and if one organism provides the essential growth factors or growth factors to or nutrients or substrates so that the growth of the other is being improved, then we say that a syntropism is being found to be involved. In the case of proto-cooperation, we have been found that most of the associations which happen in a proto-cooperation would lead into syntropic relationships in which the growth of the organism is being found to be increased. Now, if you go to consider cooperation in a single colony of a microorganism, you can see that a cooperation is usually been evidenced during an extended lag period. That is, now for example, when a very small inoculum is used for a culture, there is a failure of the growth, and this would the growth will be it will take some time, and there would be an extended lag period or a complete absence of growth would be happening when there is a very small inoculum is being present. And the semi-permeable membranes of the microbes, and they are imperfect, and they tend to leak low molecular weight metabolic substances that are being essential for the biosynthesis and growth. And we can see that within a population, there are more concentrations of these extracellular metabolites that will help them to counteract the less and facilitate the reabsorption. But in a single cell or at a very low population densities, the loss exceeds replacement rates and it will prevent the growth. So, in the case of a microbial population, when it is occurring in a colony form, what happens? The cooperation happens between various microbial cells of a colony by providing the extracellular nutrients within each other. So that is a case of cooperation which has been found in a single colony of microorganisms, which has been found during the lag period. Now, if you're going to look this cooperative reactions, it could also happen between various other organisms. Various organisms. Now, the previous example we were talking about the cooperation between a single colony of microbe. But if you're going to consider different microbes, now for example, you have your D-sulfovibrio and chromatium. They also have a cooperative symbiotic process over here. What happens is the organic matter as well as the sulfate, which has been required by D-sulfovibrio, is being provided by chromatium in its photosynthetic driven reduction of carbon dioxide to organic matter and the oxidation of sulfide to sulfate. So here, chromium is the it is the photosynthetic member, and it utilizes light and synthesizes organic matter as well as sulfate, and is being utilized, and the organic matter and sulfate is being provided towards the what? To desulfovibrio. Desulfovibrio in turn utilizes this organic matter and sulfate and releases carbon dioxide and hydrogen sulfide, which can be in turn utilized by chromatium. So these two organisms have a what? A cooperative relationship between each other in which both of them are being benefited. But the thing that we have to say here is, even if desulfovibrio is not there, chromatium can live. As well as, even if chromatium is not there, desulfovibrio can live. That is, the organic matter as well as the sulfide can also be provided by various other agents other than the chromatium. So, though desulfovibrio as well as chromatium benefit by their association, it is not an obligatory relationship for both of them to occur. So this is a kind of cooperative symbiotic process. Another example of the cooperative symbiotic process is between the nitrogen fixer Acetobacter as well as the cellulose degrader Cellulomonas. Acetobacter uses glucose provided by the cellulose degrading organisms such as Cellulomonas, and which this Cellulomonas uses the nitrogen fixed by the Acetobacter. So if you go to look into this, you can see here the cellulose, okay, the cellulose degrading organism Cellulomonas will degrade cellulose and provide glucose. And this glucose is being utilized by the nitrogen fixer Acetobacter, and this Acetobacter will in turn fix nitrogen to form ammonium, and that is being utilized by the Cellulomonas. So here also we have to make sure that the cooperative interaction between these two organisms is not a what? It is not an obligatory factor. That is, even if they are being dissociated, they can survive, or they can be viable, though their functional rate or the function of it might not be as when they are being associated. So that's a case of cooperation. I guess this is clear to you. With this, we come to an end of the cooperative as well as the proto-cooperative interactions which have been carried out by the microorganisms.